There are some kisses that leave their mark. In 1907, the artist Gustav Klimt painted the kiss in oil and gold leaf on canvas. It was a kiss that would go down in history. 110 years later, the Institute of Evolutionary Biology took part in research on dental plaque fossilized in the teeth of 5 Neanderthals. This study would lead to the discovery that Neanderthals and humans might have kissed more than 40,000 years ago. Far from the images of war, conflict, and extinction that might usually come to mind, this study allowed us to understand that there were also alliances between humans and Neanderthals, and maybe even intimacy. All this was revealed thanks to microorganisms in the mouths of the 5 Neanderthal individuals becoming fossilized in their teeth and leaving their mark in the form of DNA. The research team was able to extract genetic material from these fossilized microorganisms. After analyzing it, they discovered a wealth of information about the diet, behavior, and social relationships of these Neanderthals, including that they had shared food or even kisses with humans. The technique used by the researchers is known as paleogenomics, which consists of recovering and analyzing the DNA found in fossils in order to decipher the information it contains. It is often said that DNA is the instruction book where organisms store the information they need to function properly. However, DNA can also be read as a history book. The genome holds a wealth of information. The DNA of a single person contains the evolutionary history of our entire species. Genetic material is also a window to the past, and with the necessary techniques, we can come to discover things that would be impossible to determine any other way. For example, you might wonder how the tip of a little finger bone could unveil the appearance of our entire human population. In 2008, a team of Russian paleontologists found a small bone belonging to a mysterious hominid in the Denisova cave in Siberia. The morphology of the fossil did not provide a lot of information. After all, it was only the tip of a pinky finger. In 2010, a team from the Max Planck Institute in Leipzig analyzed the DNA of the little finger using paleogenomic techniques. They discovered that the bone belonged to a girl from a previously unknown hominid population, the Denisovans, who lived more than 70,000 years ago. The little finger was found alongside 3 molars in the cave, which also belonged to individuals from this extinct population. However, none of this evidence, neither the molars nor the phalanx, could unlock one of the greatest mysteries surrounding the Denisovans. What did they look like? In 2019, a team led by the Hebrew University of Jerusalem, with the participation of the IBE, used the latest bioinformatic techniques to analyze the DNA recovered from the finger bone and reconstruct what the mysterious Denisovans looked like for the first time. Just one finger bone was able to disclose as many as 56 distinctive features of these hominids. As you can see, the history of human evolution is one of diversity. The different populations of different human lineages coexisted, interacted, and mixed with one another. The proof of this can be found in our genes, where the fragments of Neanderthal and Denisovan DNA that we still carry today can be identified. Yet, despite the diversity of our history and populations, the DNA of all humans is extraordinarily similar. In fact, thanks to genomic sequencing techniques, we now know that we share 99% of our genetic code with chimpanzees. This means that, however different or geographically separated 2 humans may seem, in reality, the genetic differences are ridiculously minute. We have learned all this from genomics and by learning to read DNA as a history book. But of course, everything has its limits. DNA doesn't last forever. After all, it's a molecule that degrades over time. So paleogenomics can only help us with the samples up to just over 500,000 years old. This may seem like an eternity, but in terms of our evolutionary history, it is barely the blink of an eye. Some fossils from our oldest lineage date back to around 4 million years ago. So, what happens when we find fossils that have literally turned to dust? In 2019, a team from the IBE analyzed a 2-million-year-old molar. It belonged to Gigantopithecus blacki, a giant 3-meter-tall ape that lived in the forest of Southeast Asia and became extinct 300,000 years ago. The molar was too old for DNA to be extracted, as it had completely degraded. Instead, the research team extracted proteins, molecules that survive for as long as the bone they are part of. Using these ancient proteins, they were able to reconstruct some of the species' genetic information. Although the information is much more limited, it can still be very valuable. Thanks to a new technique known as paleoproteomics, the team reconstructed the ancient evolutionary history of Gigantopithecus. We now know that it was related to today's orangutan, and that these two species shared a common ancestor some 10 million years ago. If used on remains discovered in Africa, the cradle of humanity, this technique could reveal the great mysteries of our earliest evolutionary history. There are fossils more than 4 million years old in which DNA has fully degraded, but paleoproteomic techniques could reveal things we still do not know today. What's more, artificial intelligence has also opened up new horizons. Researchers at the IBE have already begun to develop algorithms that simulate the possible paths human history has followed. The goal? Finding the path that brings together the puzzle of our evolutionary history and the genetic diversity observed in modern humans. It's not science fiction, it's just science. This is known as deep learning, and it has allowed us to predict that, in addition to Neanderthals and Denisovans, there may have been a third extinct ancient population, a hybrid of the first two, that reproduced with modern humans in South Asia. In fact, this hypothesis has gained traction in recent years after the discovery of the skeletal remains of Deni, a 90,000-year-old woman, the child of a Neanderthal mother and a Denisovan father, in the Denisova cave. And Deni may not have been an isolated case, but part of a more general process of intermingling. The human past seems to be an unfathomable puzzle. A scientific discovery may provide many answers, but it can also open many more new questions. What we do know is that advances in research techniques have allowed us to continue deciphering our past, and hopefully will also be able to help us better understand who we are as a species today and who we will be tomorrow.